Good day everyone, welcome to the course on next generation sequencing technologies, data analysis and applications. In the last class we discussed SAM format, alignment section, some of the fields that are present. So, we will continue that and we will complete the SAM file format in this class, right. So, you will be able to completely understand what kind of information you have in that file. Okay. And if you are writing code, this will be very useful. You can process these SAM files and you can extract some information. For example, you want to identify the genetic variants from your mapping or other kind of information, you will be able to extract those by writing codes. Okay. So, this is the topic that we will be covering today. We will discuss the alignment section, what kind of information you have in the alignment section. These are the keywords, mandatory fields and optional fields. We have already started discussing the mandatory fields, right. So, we have discussed first 6 fields and this is where we ended in the last class, right. So, we talked about Seeger strings and we have seen what kind of letters we use to represent Seeger strings, right. So, what we will do is we will actually look or interpret the Seeger string from the real data, real output file, and we will understand what information you have in this Seeger string. Okay. So, you have these letters, but you also have some numbers, right. So, what kind of why do you add those numbers, what do they mean, we will explain when we actually go to the real data set. So, moving on to field 7, we have something called R next. Okay. So, up to field 6 you are getting alignment information for the read itself. Okay. So, the read that we are working with, the Q name, right, that information is up to field 6. Okay. From field 7 onwards, if you are dealing with paired end data, it will give you information about the mates alignment. Okay. So, you have read 1, read 2. If you are looking at read 1, up to field 6 you will get alignment information for read 1, but from field 7 onwards you will have alignment information for read 2. Okay. So, the R next means name of the reference sequence where mates alignment occurs, right. So, similarly to similar to the this ref NC some number, right. So, here you will also get some reference sequence name or ID, right, that will be in field 7. So, this will be actually set to equal to sign, right. So, this will mean if the mates reference sequence is same as the reads reference sequence, right. So, if this reference sequence name is same as R name, right, this is the reference. So, they are mapping to the same chromosome, right, or same reference genome, right, that kind of information will be given here. So, this is set to star, right, if there is no mate, if you are dealing with single end data, this is not present. Okay. The next field is field 8, this gives you P next. So, this is again POS equivalent for the mate. Okay. So, this is giving you the position of the mates alignment in the reference track. Again, it is based on one based positioning system, right, that we just described, right, and this is again describing the leftmost characters location in the mates alignment. Okay. So, leftmost position. So, as you can see, this is kind of consistent for POS as well as for P next. Okay. And it is set to 0 if there is no mate in the data, right. So, this is if I just highlight that this is the position that you get, right. So, if you have read 1, read 2, this is the position that will be represented in P next, right, the coordinate system, right. Again, this could be some number we have seen in the real data in the last class, the POS, right, it has some number. So, here will you will also get some number. Okay. So, what about field 9? Okay. So, field 9 actually gives you something called the signed observed template length or fragment length. Okay. So, this is important, right, if you are dealing with paired end data, you want to probably generate what kind of fragment length you got from your experimental procedure, right. So, when you fragment the genome, you want to know what kind of fragment length distribution you got, and that information could be useful, right, for the next time you want to prepare an sequencing library. Right. So, here you get this template length or fragment length. So, you are dealing with paired end data, you have these fragments, these reads coming from both ends, right, and you want to calculate what is the fragment length that actually generated these two reads, read 1 and read 2. Okay. So, this is given in T len, right. So, this absolute value of T len is actually n minus start plus 1, right. So, that is very simple, right. If you have this end here, the mapping ends here, right, on the right. So, you have this n and then you have the start. So, we can take this example, right. So, that will be easier to understand, right. So, end means this is probably the end, right, this is the end position and this is the start position here on this side, right. So, this is n minus start plus 1, that would give you T length. Okay. Now, so one of the things you probably have noticed, it says signed observed template length. Right. So, we have calculated T length, right. This is a positive number, but we will also have to have a sign. Okay. So, there is a plus sign, minus sign, and we will understand why we need a sign. Okay. Because we are dealing with paired end data, one of the reads will get a plus sign, the other read will get a minus sign for the same T length, right. This is important to actually figure out which read is occurring upstream of which read, right. So, whether read 2 is upstream of read 1 or read 1 is upstream of read 2, right. That information you can gather from this sign of T length. Okay. All right. So, as I said, right, in case of if you have this situation, right. So, you have this absolute value of T length, right. So, this actually how do you calculate from this P next and POS, right. So, if you are looking at, let us say, read 1 alignment, you want to calculate the fragment length. So, how do you go about it? So, remember this POS gives you the leftmost position here and P next gives you the leftmost position here, right. So, you get this absolute value of P next minus POS plus the length of read 2, right. So, here you get the leftmost positions in the coordinate system, right, and then you have to add the length of the read 2. So, based on these sum of these fields, you can calculate the T length value. Okay. So, in case this is interchange, right, you have this read 2 upstream of read 1, this then it will be replaced by length read 1. Okay. So, this part remains the same, absolute P next minus POS plus length of read 1. Now, in case of Illumina, you are working with let us say read 1, read 2 data which are of same length, then it does not matter, you can use read 1, read 2, but also be careful, right. If you are working with stream data, the length of read 1 and read 2 may not be same, right. So, in that case you have to again check carefully. Now, what about sign? How we actually give the sign? So, sign is very simple. So, you give positive sign for leftmost segment. So, if read 1 is upstream of read 2, right, this is the leftmost segment. So, you get positive sign. For the rightmost segment, the read 2 is downstream, then the read 2 will get the negative sign. The T length value, the absolute value remains the same, you simply will change the sign. Okay. So, this figure illustrates, right, how you generate this plus sign and minus sign. So, read 2 in this case is upstream of read 1. So, this is the leftmost segment. So, this gets positive sign for T length, right. So, if let us say T length is 300. So, for read 2 in the T length field you will have plus 300, right. So, or simply 300, you do not say plus 300. For read 1 in the T length field you will get minus 300, right. So, as we go into the real data, you will actually see you will be able to see these signs. Okay. All right. So, this value is actually valid only for paired end data, right. So, if you are dealing with single end data or unmapped pair, you cannot generate this fragment length, right, because you do not know the fragment length. You just, if you are dealing with just sequence data from one end, so the value is set to 0. Okay. So, the final two fields actually field 10 and field 11, right. So, field 10 is actually very simple, this is the segment or read sequence. Okay. So, the read that you are working with is your read sequence. Okay. Again, going back to the first part, right, when I mentioned, right, everything is given with respect to the forward reference strand. So, if a read is mapping to the reverse strand, right, you will get the reverse complement sequence in this field, right. So, SEQ will give you the reverse complement sequence. Okay. So, this you need to keep in mind. Okay. Now, if the sequence is not stored, then you get set to, this is set to star. And the final field, right. So, this is the quality score, again this is same as the Fred score that we get from the FASTQ file. Okay. So, this is an ASCII encoded simply copied from this FASTQ file, right, and this is in Fred plus 33 encoding, right, the new Illumina data. For older Illumina data it will be Fred plus 64. Okay. And this is set to star if the quality score is not stored. So, sometimes you may not want the sequence or the quality score in this SAM file, right, because that is simply just increase the size of the file, and this information is already present in the FASTQ file, right. So, may not require these fields. Okay. So, just to summarize the compulsory fields that we have discussed so far. Okay. These are the just a brief summary what you will get, right. So, you have Q name, which is a string, right, this is a name for the read. you have flag, this is an integer number, right. So, you can expect some number, right. We have seen this is a sum of the applicable flags. You have something called R name, which is again a string. This is the reference name, right, reference sequence name. It could be genome or chromosome number, ID, name, right. Then you have the fourth field, which is the POS, right. This is an integer, right, and again we are following this one based positioning system, and this is the leftmost position, right. We have discussed again. The fifth field is map Q. This is the mapping quality. This gives you probability of error in the alignment, right. And then finally, field 6 is the Seeger string. Again, we have seen at least described how this Seeger string is derived, right. What these letters mean, you have described some letters like M, I, D, H, S, etc. Then you have field 7, which is the R next, right. This is describing these two fields, next two fields, they are describing the alignment of the mate in case of paired end data, right. So, if you have single end data, you will not get any value, right. So, R next gives the string. So, this is set to equal if it is same as R name, right. So, R next and R name they are equal, then you will get this equal sign. Then P next is the position, right, of the mate in the reference sequence, and we can derive the fragment length, which is given in the 9th field, right. So, this is T len. Again we have described how we actually derive this T len value. And final two fields, these are simply giving the sequence of the read or reverse complement and the quality score that comes from the first Q file. Okay, so, what we are going to do is we are now going to go into the file, and we are going to check these fields, right, and then how they look like. Okay. So, let us do that, and then beyond that we will discuss the optional fields that are there and what kind of information you get from the optional fields. Okay. So, let us now go to the terminal into that file. Okay. So, here is the file again, right. So, we have come back to this output file, the output.sam, and we have described already up to this point, right. So, this field here 44, this number that you see is describing the mapping quality, right. The next one, as I mentioned, the field here is the Seeger string. Okay. So, you can see this 65M, 65M, and then you have 125M, etc., and sometimes you will see this 2S, 123M, etc. So, as we have described some of these letters, right, M, I, H. So, M means match. So, match means there is a read, right, there is no insertion or deletion, there is a base for every base in the read. So, what do we indicate by these numbers 65 or 125? Okay. So, 65 means you have 65 base match. Okay. This could be match or mismatch, again we do not count that here, we cannot get that information here. Okay. So, 65M, that is good because we have like the full alignment for this read that we have, right. So, this information tells us okay, the full read has been aligned against the reference sequence. 125M means we have 125 bases in the read sequence that has aligned against the reference sequence, and there is no insertion or deletion. Okay. And these are ideal scenarios, right. So, if you have very good quality data, most of these will be M's, right, you will not see anything else most of the time. But then here you have these 2S 123M. So, what is this S? So, S stands for soft clipping, right. So, soft clipping means these bases were clipped from alignment. So, they were not aligned, okay, but they are present in the read sequence in the SAM file. Okay. In the read sequence they are retained. Okay. So, what this means now, these numbers 2S 123M. So, 2S means first two bases, right. So, we start, right, first two bases were soft clipped, they are not used for alignment, and then followed by we have 123 base match. Okay. So, this Seeger string actually signifies that. Okay. So, these 2S or S will appear if you are going for local alignment. You will see a lot of this S signs, means there will be a lot of soft clipping because you are going for local alignment. Okay. So, you can continue, right, and again you can interpret this one 1S 124M, it means we have first base soft clip, then the next 124 bases have been aligned. Okay. So, I think this is clear now. So, let us look at a bit more complicated examples, right. So, here for example, you have this 46 M, right. You can see I am highlighting with my pointer. So, you have 46 M, 1 I, 78 M. Okay. What does this mean? It means first 46 bases are matching perfectly with the reference, then you have one insertion, okay, and then you have 78 bases matching again. Okay. So, you have insertion in between, and on one side you have 46 matches and on the other side you have 78 matches. Okay. This is what this figure tells us. Okay. And similarly, you can actually search a bit more and you can see a more complicated scenarios. For example, here again this is slightly more complex one. Here you see 113 M, 2 I, 1 M, 9 S. Okay. So, again following the same kind of explanation. So, 113 M means 113 base match, then you have 2 insertions, then again we have 1 match and 9 bases soft clipped. Okay. So, what you see this towards the end of this read, you have this insertion, 1 match and then 9 soft clip, means this quality of alignment probably has dropped a bit. So, you would have to actually check very carefully the alignment here and probably want to discard this last part, right. So, whether this alignment actually has worked or not. These 2 insertions they may not be real insertions, because you have this 9 soft clip, means these are not matching. So, program could not find any match. So, it is removing them, right. So, you would have to be a bit careful when you are dealing with this read alignment. Okay. So, maybe we can search for another example. I will search so that I can show you, right, how this will look like. Let us search. I think we will see if we can find some D. Okay. So, here are some nice examples, nice complicated ones. Okay. So, here you see, right, you have 26 M, 2 D, 50 M, 49 S. Okay. So, this contains a lot of these different signs, right. So, 26 M means first 26 bases they are matching, then you have 2 D means 2 base deleted, right. So, these are present in reference, but not present in the read. Then you have 50 M, right. So, 50 bases matching again, then you have 49 S, 49 bases soft clipped. Okay. Now, this 49 S should ring a bell, right. This is a huge number of bases that have been soft clipped. So, maybe this read is there is some issue with this mapping here. Okay. Similarly, the next one also is quite complex, right. 35 S, 28 M, 2 D, 62 M. Okay. So, 35 S, 35 bases soft clipped, 28 matches, 2 deletions, 62 matches. Okay. And so on, right. So, you can probably find lot more examples. So, here is a nice example again. You have 120 M, right, 2 D, 1 M, 4 S, right. So, you will see like most of the cases, most of the situations you will have these only M. So, that is good, you do not have to interpret anything, but the reads that have that contain insertions or deletions, you can identify them very quickly using those Seeger strings, right. So, if you have Seeger strings I or D, you can say okay, these are the reads that contain deletion or insertion, right. So, this is quite helpful if you are trying to working with indels only, or maybe you want to filter out indels, you are not interested in indels. In that case you can use this information. Now, here I will point you towards some data here, right. You can see these 2 lines, right, these are part of the same fragment, right, and some of them are given as star 0 0 means there is no alignment, right. So, star means there is no position, there is no sequence, right, name. So, it could not map these reads to the reference sequence. Okay. So, now this Seeger string is clear. The next field is the reference name for the mate. Okay. This is the R next, right. So, this is set to equal means the reference is same, right. So, it is mapping to the same chromosome. So, that is why this equal sign. Okay. Instead of writing this again, you have the equal sign. Then followed by you have this position P next, right. So, this is comparable to POS, right. So, you can see these 2 reads here, again these are paired in data, okay, and you can see these are actually mapping to the same positions, right. So, here is the position of the mate 2, okay, of this read. So, the other mate, so this is the position. We can take some other examples. For example, we can go down here, right. For example, we can take this one. Let us say this is we have 125 M matches, right. So, again this equal signs for most of these, right, meaning they are mapping to the same reference sequence. The mate is mapping to the same reference sequence as the read. Then you have the positions given, right, of the mate where this is aligning, and then you have this T length. Okay. So, this is the fragment length. Okay. Now, for this one, this is negative, means this is occurring downstream, the mate is occurring upstream of this read. Okay. And for the mate here, right. So, as I said, the data is stored in pairs, right. So, you have read 1, read 2 data one after another. So, for the mate you can see this T length is positive, right. So, this one is the one that is occurring upstream, right, or occurring in the leftmost position, and this read here, right, this one, this mate is occurring downstream, this is the rightmost positions. Okay. So, hopefully these are clear. Now, these two fields, right, they are describing the mapping of the mate. The next field is the sequence, right, you simply get the sequence data here, right, this is what you see, and then you have the quality score as we go along, right. So, here is the quality score ASCII encoded, again simply taken from the reference, simply taken from the FASTQ file. Okay. All right. So, this actually is the end of the compulsory fields. Okay. Now, what you have after these are the optional fields, and you can probably see some of them here, right, very if you look carefully, these are actually describing the optional fields, these ASI, etc. Okay. So, we will now describe what these optional fields actually mean, and then we can again look at the data to better understand these fields. Okay. So, let us go back to the presentation and let us discuss these optional fields. Okay. So, going to the optional fields, right. So, all optional fields, they are given in this specific format. Okay. So, we have something called tag, you have type, then the value. Okay. So, what is tag? So, this is a 2 character string, right, and then type, this is a single letter usually, right, this defines the format of the value, right. So, whether the value is an integer, whether the value is a string, that information will be given in this type field. Okay. So, then there are specific tags that we use to describe the alignment or the mapping. Okay. So, here is the first one. So, notice carefully, this is given as AS colon. So, that is the tag, right. Then you have the I, means we can expect an integer in the value which is given here within this less than greater than sign n, right. So, here we will get a value. So, as I, so I means integer. So, we will get an integer value in place of n. Okay. So, this gives us an alignment score, right. So, and this is present if you are working with aligned reads or aligned data. Okay. Now, this alignment score, right, better score, higher score means you have better alignment or better matches, right. Then you get something called excess I n. Okay. Again, this is an integer field, right, I means integer. So, you get a number. Now, what you get in excess is the alignment score for the best coding alignment found other than the primary alignment. So, S actually is the reported alignment, right, that alignment that is reported, the reference name, the position, this is the primary alignment, right, where the alignment has happened, that gives us this alignment score AS. But sometimes reads can map to multiple positions, right. So, that is why you get these alternate alignment scores, right, or these are the secondary alignments that are reported. Okay. And this will be given when you have more than one alignment for the read in the reference sequence. Okay. So, this is something we may have to be careful, right, when we are processing this SAM file, we may have to carefully check whether there is alternate alignment, whether they are mapping to multiple positions in the genome, because that can also influence some of the downstream analysis. Then you have this YSI, right. So, this is again alignment score for opposite mate in the paired end alignment, right. So, this if you are working with paired end data, you will get this information. Okay. Then you have something called XN, okay, and the number of ambiguous bases in the reference covering this alignment. So, again these are all I means integer numbers, right. So, if you have ambiguous bases in the reference that you want to know, right. So, because in those positions we will not be able to call any mutation or mismatches, right. So, this just simply gives us the number of ambiguous bases in the reference. Okay. Now, following this, there will be this very important information that actually describes the alignment, whether there are mismatches, etc. Now, one thing you should remember, right, you may not see all these fields in the data. Again, these are optional fields. So, in some cases you may see xn, you may not see xn, right, if there is no reported value. If n is 0, this may not be reported. These are optional fields for a reason, right. So, you will see maybe some of them and not all. Okay, so, the next one is something called xmin. Again, it is an integer and it actually gives us the number of mismatches in the alignment. Okay, so, if you compare the read against the reference sequences, how many mismatches you have in the read. Okay. Then you have something called XO and number of gaps that you get for both read and reference gaps in the alignment. Okay. So, this is simply describing the indels, number of indels that you have in the data, and this is also the number of gap extensions xgin, right. So, again this is looking at both indels, right, and this is again will so taking xg and xo, that you will get an information about the indels in the data that you have. So, you can see like CIGAR will give you an indication whether you have indels, and this will give you the numbers, right, how many indels you probably have in the data. Then you have something called NMI. So, this is the edit distance. So, that is the minimal number of one nucleotide changes, right, whether it is insertion, deletion or substitution, that you need to do in the read sequence to get to the reference string. So, okay. So, it is kind of like a number of mismatches plus number of indels that gives you this edit distance, or simply how far the read is from the reference sequence, right. So, most of these cases this should be 0 because you do not have any mismatch or indel in the data. Okay. Again, we will go back to the SAM file and we will carefully look at how these numbers actually look like. Okay. Then you have something called YFZS. So, this is if there is some sort of filtering, it might indicate why read was filtered out, etc. You may not see this, right, if the read is present. Then you have something called MDZ. Okay, so, I should comment on this Z, right. So, you will Z, whenever you see Z in the second type field, right. So, this means there will be a string in the value. Okay, so, this S means string. So, you will see a string, right. So, this will be an alphanumeric. Okay. MDZS will be a string representation of the mismatched reference bases in the alignment. So, what are the changes that you see, right, that will be given in this MDZ. Okay. Finally, you have something called YTZ value, right. So, again this is a string, you will see some of these options. So, only one of these, right. So, you can have UU, right, this read is not part of a pair, if you are dealing with single end data, this will be happening. If you have CP value, right, the read was part of a pair and then they align concordantly. So, this is referring to concordant mapping, right. We described what is concordant mapping, and most reads will show concordant mapping. If you see YTZ DP, means this is a discordant mapping, right, and perhaps we want to look at those data carefully, and if there is an indication of structural variation or gene fusion, etc., those will be found with this discordant mapping. Then you have UP value, right. So, the read was part of a pair, but the pair failed to align concordantly or discordantly, right. So, this is something that again means you have single mapping, right. So, only one of them aligned. So, they did not map as a pair. All right. So, just to final illustration, right. So, this is the Q name that you will get. Then you have something called flag. This is a flag. This is the R name, right. Then you have this POS field. This is the map Q. This is the CIGAR. Then you have R next. This is P next. Then you have the TLEN. Okay. So, these are the part of the compulsory fields. You have sequence and quality, of course. These are also present in the data as part of compulsory fields. Then following that you have these optional fields, okay, and you get these different characteristics, right. So, that describe the alignment. Okay, so, what we are going to do, we will go to the file now, the terminal, and look at the actual data, actual output data, and see how these look like, these optional fields. So, we have seen up to the compulsory fields. So, we will now check how these optional fields look like in the real data. Okay. So, let us move here, right. So, you see this here, okay, and you can see these numbers, right. So, you can see ASI 127, this is the alignment score. There is no excess, so means there is no alternate alignment found. So, this is a unique mapping. Then you have this XMI, right. So, XNI is 0, there is no ambiguous base in the reference, you have xmi 1 means you have one mismatch, xoi 0, xgi 0 means there is no gap, right, no indel, right, in the data. NMI is the edit distance. So, what will happen is that xm plus xo plus xg, these numbers will give you the nm. Okay. Then you have description of this mismatch that is found, right. So, if you carefully look at this, so this is 30T 36, right. So, that means you have this so the length is 67 here, right. If you come to the same read data, we just go into this field, it is 67 and that MDZ means 30T 36. Okay. So, 30T means you have 30 bases that are matching exactly, then you have a T mutation, right, and then you have 36 base match. Okay. Then you have YSI, we have described what is YSI, and then we have this YTZ. This is again quite important. So, this MDZ, this NMI, these are actually the important optional fields that you often look at if you are processing these files ourselves. Okay. And YTZ CP means you have concordant mapping. Okay. So, we can actually also see some of some reads, maybe you will find this discordant mapping here, for example, here I see, right. So, in here you can see these two reads, right. So, you have these YTZ DP reads, right. So, they are actually showing discordant mapping. Okay. So, maybe we can also find out the reason why they are showing discordant mapping, whether mapping to two different reference sequences or two different reference chromosomes, right. So, the chromosomes are reference sequences here, or they are mapping too far away from each other, right. So, here actually this they are mapping to the same chromosome. So, these are the two reads here, right. You can see they are mapping to the same chromosome because of this equal sign, but they are too far from each other. So, 843. Now, the default option is 500. So, maximum distance that is allowed is 500 in BOTI2. So, you can of course change this using this minus x option when you run BOTI2. So, because this distance is too high according to the cutoff or threshold that is set in BOTI2, it is calling these as discordant mapping. Okay. So, I think this is clear now, right. What are the information that we will get in these fields, and some of this information could be very useful. For example, in here for this read data, you can see you have again a quite elaborate information and multiple mutations that are present, and you will see this will translate into NMI or XMI equals to 6, right. So, NMI is just a sum of XMI, XOI and XGI, right. So, XMI 6 means there are 6 mismatches. Okay. And in some cases, for some reads you can see this excess field, right. So, you have this ASI, XSI, and in this case what you see is that alignment score and the alternate alignment score, they are actually same. Okay. And in these cases you would have to be careful, because this means they are mapping to repeat sequences, right, and the alignment score is exactly identical, right. So, that means they are showing probably exactly same alignment. It is not like one alignment is better than other, they are probably of similar number of mismatches, etc. All right, I think we can now conclude. So, what we have, so this is the reference we have seen, and to conclude we have actually seen that SAM file contains header section and alignment section. For each alignment, the SAM file shows the location of alignment, the quality of mapping, as well as mapping of the mate if you are dealing with paired end data. Each alignment line contains 11 mandatory fields, and there are also optional fields, and these optional fields are also quite important, right. They give out certain important information about the alignment, and these each alignment also report mismatches in one of the optional fields. So, this actually is important if you are trying to look for genetic variants, etc. That is it for today. Thank you.